So we've just finished talking about the inheritance of genes on chromosomes, and next we're going to shift to this idea of the central dogma. So the genetic information that flows from DNA to RNA to proteins. And we're going to start by focusing on DNA to RNA. So first we'll ask, what do genes do? And then examine the central dogma of molecular biology. We'll then jump to chapter 17 and talk about how DNA is transcribed into RNA, and then jump to chapter 18 and talk about an instance where genes are regulated, and so they're turned off and on in different conditions. So that's what we're going to talk about for this lecture. So biologists knew about DNA, heredity, chromosomes, and genes, but they did not understand gene expression. And what I mean by gene expression is turning DNA into functional molecules. And some really important experiments by Beadle and Tatum, and here they are receiving their Nobel Prize for their work, really mapped out how genes work. And Beadle and Tatum designed these quite simple experiments that were quite elegant. And Beadle's thought was one ought to be able to discover what genes do by making them defective. And so this is the experiment that they set out to do. So in their experiment, they attempted to knock out a gene by damaging it and then observe the phenotype of the mutant. And so when they were able to damage particular genes, this created loss of function alleles. So these are alleles that do not function properly. And they used the model system Neurospora crassa, and this is a bread mold. And to damage the genes, they irradiated the cells, and this induced point mutations in the cell. And they were waiting to find these mutants that didn't function properly. And so they were trying to observe if there was a defect in a particular organism on their ability to grow or produce a particular compound. And so what they saw was, for one example, is the inability to produce a compound needed for growth, and that was vitamin B6. And this was due to a defect in a single gene. They also noticed the failure to produce other compounds, and that was due to defects in other genes. And so this inspired their one gene, one enzyme hypothesis. And just here are some of the illustrations from their paper. These were drawn by Beadle, mapping out the mutations and whether or not they could grow in different medium. And the mutants that they made could not make compounds because they lacked the enzyme needed to synthesize it. And so that was their observation that supported this hypothesis. Serb and Horowitz then took this a step further by studying this three-step metabolic pathway that produces arginine. And they set forth to do these experiments that we call genetic screens. And they were testing the one gene, one enzyme hypothesis. And they performed a genetic screen by growing mold cells on medium that lacked arginine. And these mold cells, these bread mold cells, had been treated in the same way with radiation to induce point mutations. And the cells that died came from a colony that was missing an enzyme from that pathway. So real quickly, I'll just explain this metabolic pathway for producing arginine. So arginine, or cells take in a precursor. Enzyme 1 then takes that precursor to generate ornithine. Then a second enzyme comes along and converts ornithine into citrulline. And then enzyme 3 converts citrulline into arginine. Okay, so this metabolic pathway will be important for their genetic screen. So what they did in their genetic screen was that they, again, produced mutants, and these mutants would be unable to synthesize arginine. And so they took these mutants and they put them on this growth medium. And here's the Neospora crassa. And here there's no supplementation. So they don't have any ornithine, citrulline, or arginine. Okay. And their experimental setup was to find cells that cannot synthesize arginine, but they grew them on different medium. So they grew them on this non-supplemented medium supplemented with just ornithine. So here's where ornithine is in that pathway. Medium supplemented with citrulline, or medium supplemented with arginine. And so just as an example here, if a cell was missing this enzyme 1, for example, it wouldn't be able to grow on this non-supplemented media because it can't convert the precursor into ornithine. But it would be able to grow in media containing ornithine, right? And that's just true for enzyme 1. But they didn't know about these enzymes at the time, and so this helped determine that there were these different mutants, okay? So this first mutant was ARG1, and it grew in that ornithine medium, and of course also in the citrulline and arginine. ARG2 was another gene identified, and this did not grow in the ornithine, but it grew in citrulline media. And so this suggested that this enzyme mediated this second step in the pathway, converting ornithine to citrulline. And then finally, a third mutant was identified. This did not grow in the citrulline media, but did grow in the arginine media, suggesting that this enzyme, this mutant, would normally function to convert citrulline to arginine. So again, if a cell couldn't grow with arginine, but failed to grow, if a cell could grow with arginine, but failed to grow without it, they concluded that it couldn't make its own arginine. And this work showed eventually, altogether, that it supported the one gene, one enzyme hypothesis, and then combined with later work showed that genes contain all the information for proteins produced by an organism, right? So these were all the proteins, not just enzymes. And so consequently, the one gene, one hypothesis was modified to the one gene, one polypeptide hypothesis. And so that's where we stand today. So how does that fit into the central dogma? So the central dogma was proposed by Watson and Crick. And here's a picture of Crick giving a talk at Coldwater Harbor Symposium. And if you can squint, you can see behind him, there's the central dogma, DNA. He's standing right in front of mRNA, and then you can read protein. And so he said, kind of a confusing statement, but the central dogma, once information has gotten into protein, it can't get out again. Information here means the sequence of amino acid residues or other sequences related to it. So he's talking about how protein gets how DNA gets communicated into protein. Crick went on to propose that sequences acted like a code, and that DNA was the information storage molecule, and different combinations of the bases could specify the 20 amino acids. And he compared this to the Morse code, which is a series of dots and dashes that encode for the 26 different letters of the alphabet. And he also suggested that the information in DNA was not directly translated into amino acids, that there was some sort of intermediate. Jacob and Monod came along later and suggested that RNA links genes in the nucleus to protein synthesis. And they coined the term messenger RNA because it was taking the message, this information message, from the nucleus to the cytoplasm where the protein machinery was located. And then other experiments also showed that an enzyme called RNA polymerase was responsible for copying DNA into mRNA. So this brings us to the central dogma, and I showed you this on the first day of class, but we have DNA, which is found in the nucleus, being transcribed into mRNA. That mRNA exits the nucleus, is picked up by a ribosome, and it's located in the cytoplasm. And at this point, the mRNA is translated into protein. Another way to shorthand write the central dogma is DNA, arrow, RNA, arrow, proteins. And the genes are stretches of DNA that code for the proteins. So DNA sequence codes for RNA sequence. That's the intermediate there. And then the RNA sequence codes for the sequence of amino acids. So that's what's happening here. And we're going to talk more about this particular step, but also what's happening here in this information flow represented by these arrows. Okay, so the roles of transcription and translation. So we describe the process of using this DNA template to make complementary mRNA. We describe that as transcription. And so this is making a copy of the information, like transcribing. If you were a transcriber or a court reporter, you would just copy down what people were saying in a court of law, right? That would be transcription. Same language. And then translation is the next process. And this is the process of using information in mRNA to synthesize proteins. The way I like to remember this and the way the textbook suggests is that we're now translating the language of nucleic acids into the language of proteins, which is amino acids. So translating meaning changing the language like a Google Translator. And so hopefully that little tip will help you remember these different terms for these different processes. Okay, let's take this a step further and think about how the central dogma links genotype to phenotype. So another way we could write the central dogma is that DNA is our genotype, and that can be transcribed into mRNA and translated into proteins, and the proteins make up the organism's phenotype. Okay, so we're going to take a look at an example here with these mice strains, okay? So recall that alleles of the same gene differ in a DNA sequence, okay? So here is the DNA sequence of a particular protein in mice called the melanocortin receptor, okay? And you can see that the melanocortin receptor starts with a guanine here in A. Okay, that is transcribed into RNA. And you can see here that this mRNA is complementary and anti-parallel to the DNA strand. And then this RNA encodes for a particular amino acid. And we'll talk more about this code next week. So when we have the sequence CGC, we end up starting with an arginine. And that encodes for a functional melanocortin receptor, and that turns this mouse black. They have dark coats. And this isn't particularly useful for the mouse because this can make them very easy to find because of predators. But here is a mutation that occurred in this DNA sequence for this receptor in which the G has been mutated to an A. And then the complementary mRNA now begins with a U. And this starts with a cysteine, which means that this receptor is no longer functional. And so these mice can't produce that coat color. And so they're white. And this is beneficial to this physical trait is beneficial to these mice in this environment. So this is the mainland mouse, and it does pretty well on the mainland where it's dark, but the wild mouse can really flourish on the beach because it blends in with the sand. So again, proteins produced by different alleles can differ in their amino acid sequence, and that's demonstrated here, right? So this is how we take genotype and really think about converting that into phenotype. So that's it for this video, and you should definitely watch the animation next on bacterial replication, and then I'll touch on it just a little bit more in the next video.